Welcome back to the Deep Dive, where we take complex, cutting-edge research and distill it into essential, actionable knowledge for you. Today, we're undertaking a deep dive into the fascinating, complex, and frankly, rapidly evolving world of stem cells. Specifically, we're going to look at how these fundamental building blocks of life are being used to repair, reconstruct, and regenerate the incredibly intricate tissues of the head and neck.
It's a field where the margins for error are razor thin. I mean, when you think about the head and neck region, it's a crossroads of function.
Absolutely. It houses everything.
It does. Organs for sight, smell, taste, breathing, swallowing, and, crucially, communication. Maintaining functional integrity after disease or trauma is absolutely paramount.
And that's always been the challenge, right? Historically, surgical reconstruction for tissue abnormalities, say from cancer or an injury or developmental defects, it's been severely limited.
It has, limited by a lack of suitable autologous tissues.
Okay, let's break that term down. Autologous, that just means tissue harvested from the patient's own body. So the essential constraint has always been you just run out of quality source material to patch the patient up.
That's it in a nutshell. You may have the surgical skill, but if you need a new segment of windpipe or a functional patch of vocal cord, you can't just keep harvesting rib cartilage or muscle indefinitely. It causes its own problems.
The research we're unpacking today, however, signals a massive shift. I mean, this is a true paradigm change. And it's driven by these astonishing advances in what we call in vitro, in a dish, and in vivo, in the body, tissue expansion. All of that coupled with some really sophisticated tissue engineering techniques.
And the core discovery that underpins all of this, the real bedrock of the entire field, is the understanding that stem cells, in all their diverse varieties, are the cellular materials ultimately responsible for all normal tissue generation, maintenance, and regeneration.
Throughout our entire lifespan.
Throughout our lifespan. So if we can isolate them, expand them, and crucially direct them, we can essentially manufacture the missing tissues outside the body or guide the body to repair itself with unparalleled precision.
So our mission today is really twofold. First, we need to create a clear glossary, outline the different varieties and common features of stem cells, just so we master the essential terminology. Then we're going to explore their current, often revolutionary clinical applications, specifically within this complex domain of otolaryngology, which is head and neck surgery.
And we have to conclude by addressing the other side of the coin, the profoundly critical role these same cells play in malignancy, the concept of cancer stem cells, or CSCs.
Right. It's a story of creation and destruction, all driven by the same fundamental cell type.
Precisely.
The scale of current research tells you how relevant this is. You mentioned a staggering statistic before we started.
I did. Yeah, just to give you context on the explosive rate of discovery here, if you search the term stem cells in the academic literature, you find that more than 20,000 related papers were published in 2015 alone.
20,000 in 1 year.
In 1 year, and it's only accelerated since. The field is not just advancing, it's accelerating exponentially. This isn't theoretical, it's the future of how medicine approaches repair and healing. And it matters immensely to every surgical discipline.
Okay, let's start at the very beginning. When we talk about a stem cell, what makes it biologically distinct? What are the essential properties that give it its unique power compared to, say, a mature skin cell?
We can really define the power of a stem cell by 3 essential properties. These are the non-negotiable criteria.
Okay.
The first is the ability to divide persistently. Unlike most mature cells in your body that divide a fixed number of times and then die, a phenomenon known as senescent stem cells are essentially immortal in the lab. They're capable of indefinite division.
They just keep going.
They just keep going. The second property is the capacity for self-renewal. This is key. When a stem cell divides, at least one of the resulting daughter cells must retain the full characteristics of the original stem cell.
So it makes a copy of itself.
It makes a copy of itself to maintain its own necessary population, ensuring that the pool of stem cells never runs dry.
And the third property must be their actual function for the body, which is creating replacement tissue.
Yes. The third, and arguably most critical property for any clinical application, is the ability to produce cells that mature into specialized functional tissues. We call this differentiation.
Right.
A stem cell division must ultimately result in cells that stop being stem cells and instead become functional components like muscle cells or skin cells or nerve cells.
So persistent division, self-renewal, and differentiation. That's the rule book. Let's look at the first major class, adult tissue-specific stem cells, also known as somatic stem cells. Where did we first truly prove these cells even existed?
Well, the recognition that many tissues continuously renewed themselves is old, but the scientific proof of the stem cell concept really solidified about 50 years ago. It came from detailed studies on cell division within the bone marrow.
Mm-hmm.
Researchers observed that while a large number of blood-forming cells could divide, the power of persistent long-term division, that self-renewal capacity we just talked about, was restricted to a very small, very specific subset of cells.
And that restricted subset became the gold standard for defining a somatic stem cell. Give us the classic example. Hematopoietic stem cells, or HSCs.
Hematopoietic stem cells are the perfect paradigm. They're the stem cells that form blood and immune cells. Researchers learned how to identify these cells by the expression of specific cell surface markers, basically proteins acting as tags on the cell's exterior.
So you can pick them out of a crowd.
You can physically sort them. They also demonstrated that these HSCs were clonogenic in vitro, which is a fancy way of saying a single isolated cell could start an entire colony in a laboratory dish.
But the real clincher must have been their function in vivo, in a living system.
Absolutely. The defining functional test, the one that proved it all, involved transplanting these cells into mice whose entire bone marrow had been destroyed by irradiation.
So blank slate.
A completely blank slate. And the HSCs had this astonishing ability to completely restore the entire blood-forming system, the full spectrum of blood cells in those irradiated recipients.
Wow.
This test unequivocally proved that these immature cells could indefinitely divide both to renew themselves and to produce all the functional, mature cell types needed by that tissue system.
That really confirms their systemic power.
Mm-hmm.
Now let's explore the spectrum of cells they can generate, which is defined by this term, potency.
Potency describes the range of differentiation. So when a stem cell can generate cells entering several different lineages, we classify it as multipotent.
For multiple.
Exactly. Our hematopoietic stem cells are multipotent. They don't just make one type of cell, but this diverse range of cell types found in blood, including erythrocytes, that's red blood cells, inflammatory cells, and numerous immune cells.
And on the other end of that spectrum, we have those that are more restricted.
That's the unipotent category. These stem cells normally produce cells of only a single lineage. For instance, epidermal stem cells typically only make new skin cells.
Okay, that seems straightforward.
But this is where the complexity comes in. Even these unipotent cells can demonstrate a degree of plasticity under specific high-stress conditions, like during a significant wound-healing event.
So they can break their own rules if they have to.
They can. They might be temporarily capable of generating cells outside their normal single-lineage mandate, adapting to extraordinary repair demands.
So even the strict specialists have a degree of biological flexibility when truly challenged. Given this, what are the major advantages of sticking with these adult somatic stem cells when you're designing a clinical therapy?
The clinical advantages are substantial, making them the default approach whenever possible. First, they are generally readily expanded in vitro. We can grow a lot of them from a small sample.
Okay.
Second, they usually maintain a stable differentiation pattern, which is crucial because it significantly reduces the risk of unexpected or unwanted tissue growth when they're implanted. They're predictable.
And the third point, which you can't overstate in transplantation medicine.
The third point is vital. Since they can be expanded from autologous tissue, the patient's own cells, they initiate absolutely no immune rejection.
Which means no need for those heavy immunosuppressant drugs.
Exactly. That eliminates the need for lifelong immunosuppression, which is a massive win for the patient.
So what are the established routine uses of these cells today?
We use donor hematopoietic stem cell transplantation routinely for treating blood cancers like leukemia. That's a standard of care.
Right.
Beyond that, the use of in vitro generated autologous sheets of epithelial cells is a well-established successful treatment. We see this used for grafting onto severe burn patients to replace lost skin, and for the repair of corneal damage to restore sight. The foundation of regenerative medicine is built on these successes.
That brings us to a limitation, though. If we need a tissue that an adult somatic stem cell can't readily produce, or if we just run out of that autologous tissue, we have to look for cells with greater inherent potential. So let's shift our focus to the next major group, embryonic stem, or ES, cells.
The discovery of embryonic stem cells about 20 years ago was an undeniable scientific watershed, but it was also the beginning of a significant ethical debate.
And where do they come from?
ES cells are isolated from the inner cell mass of blastocysts. This is the small cluster of cells that would normally go on to form the actual embryo during development.
What makes them so powerful biologically? What's the key property?
They possess pluripotency. This is a higher degree of potency than the multipotency we just discussed.
Pluripotent. Okay, so what can they become?
Pluripotent cells can differentiate into any cell type derived from the three primary germ layers, ectoderm, mesoderm, and endoderm, which means they can become virtually any cell in the adult body. They can be isolated, greatly expanded in the lab, and then differentiated into multiple cell lineages.
How do researchers even guide that differentiation process? It sounds like magic.
It's highly controlled science, really, designed to mimic nature. Differentiation is directed in vitro by providing very specific signals, like growth factors and chemical cues, that essentially mimic the tissue interactions and signaling pathways that normally guide the emergence of specific lineages during natural embryonic development.
So it offers scientists an essentially unlimited source of differentiated cells relevant for replacement therapy.
An unlimited source, yes, that's the potential.
An unlimited source is certainly the ideal, but you mentioned the complexity. Given their vast potential, why aren't ES cells the standard for all regenerative procedures right now?
There are two main hurdles that have severely limited their widespread clinical use. The first, which really defined the early research landscape, is the strong ethical opposition.
Right.
Because generating ES cells involves the necessary destruction of living human blastocysts, this process provoked significant moral and political opposition, which placed constraints on research funding and scope in many jurisdictions.
And the second challenge is purely biological, dealing with the body's defense mechanisms.
That's the challenge of immune rejection. Since the tissues produced from ES cells express histocompatibility antigens determined by the donor embryo, they are not genetically matched to the recipient.
So the body sees them as foreign.
It sees them as foreign and attacks them. This restricts compatibility unless the tissues are transplanted into specific immune-privileged sites like the eye, or unless the recipient receives heavy, often risky immune suppression. Or, and this is highly experimental, the stem cells themselves are genetically modified to reduce antigen expression.
So we have the adult somatic cells offering safety but limited replacement capacity. And we have embryonic stem cells offering unlimited potential but high rejection risk and ethical complexity. This biological dichotomy begged for a solution.
It did.
That, I imagine, is what drove the breakthrough leading to induced pluripotent stem cells, or iPSCs.
You've perfectly summarized the scientific pressure. This discovery was arguably the most transformative breakthrough in modern stem cell biology. Researchers studied ES cells intensely to understand how they maintain their youthful, potent state.
What kept them so versatile?
Exactly. This led to the identification of specific transcription factors. These are genes associated with maintaining that state of stemness.
And what were those key factors, this sort of blueprint for pluripotency?
The primary factors identified were a group of four: OCT4, SOX2, CMYC-X, and KLF4. Now, to understand the breakthrough, you have to understand why these four are so powerful. They are transcription factors, meaning they are proteins that control the expression of thousands of other genes.
So they're master switches.
They are master switches. By activating this core group, they essentially override the genetic programming of an adult cell.
So the breakthrough wasn't just finding the genes, but finding that you could force a mature, differentiated cell, say a simple skin fibroblast, to express these genes.
Exactly. The key was that the induced expression of these specific stem cell genes in an already differentiated cell resulted in those cells acquiring stem cell properties remarkably similar to ES cells: indefinite division and, crucially, full pluripotency.
That fundamentally proved that cellular differentiation, which we previously thought was a one-way street, was in fact reversible. It's like hitting the biological reset button on an adult cell.
It demonstrates a degree of cellular plasticity that was thought entirely infeasible just decades ago. And the clinical implication is immense.
What is?
This offers the possibility of generating pluripotent cells from autologous adult tissues. This essentially sidesteps both the ethical issues associated with ES cells and the issue of immune rejection, offering the best of both worlds.
Were there immediate concerns about safety, given that you're using genetic alterations to start this process?
There absolutely were, yes. The initial concerns centered on the potential for carcinogenic effects, especially because one of the original factors, CMM, is a known oncogene, a gene associated with cancer.
A huge red flag.
A huge red flag. The necessary genetic alteration used for induction raised significant concerns. However, research has largely mitigated this by developing newer methods for induction. These modern techniques specifically avoid permanent genetic alteration using transient delivery systems or small molecules, making the resulting iPSCs much safer for potential clinical use.
The value of iPSCs isn't just about making new tissue either. They seem incredibly important for basic research.
Oh, they're invaluable non-clinical tools. If a patient has a rare genetic disease, researchers can take their readily available skin cells, convert them into iPSCs, and then differentiate those iPSCs into the specific disease cell type, say, cardiac or neuronal cells, and study the disease process in a dish.
A disease in a dish.
It's exactly that. This allows for unparalleled modeling of genetic diseases and, crucially, provides a platform for high-throughput assays of drug effects before you ever test a treatment on a patient.
That brings us to our final major type in this section, mesenchymal stem cells, or MSCs. Where do they fit, both physically in the body and functionally?
Mesenchymal stem cells are incredibly flexible workhorses. They are multipotent stem cells that can be isolated from a wide variety of adult and fetal tissues, most commonly bone marrow, but also fat tissue, dental pulp, and umbilical cord.
So they're everywhere.
They're thought to be broadly distributed in many adult connective tissues, yes.
What are their signature characteristics that make them so popular in head and neck regeneration?
They are typically fibroblast-like cells, meaning they resemble the structural cells found in connective tissue. They have excellent expansion potential and strong differentiation capabilities. But what makes them unique for clinical application is a dual function. They typically differentiate into structural components: osteoblasts, which are bone cells, or chondrocytes, which are cartilage cells, and they possess powerful immune-modulatory properties.
Immune-modulatory properties. How does that translate clinically? What does that mean?
It means they can actively influence the local immune response. They often reduce inflammation and prevent excessive scarring or immune flare-ups at a site of injury or graft. They're like on-site paramedics.
While they are known for bone and cartilage, have they shown any of that plasticity we discussed earlier?
Yes. While their primary differentiation pathway is mesodermal, so bone, cartilage, fat, there have been compelling reports suggesting greater plasticity. This includes their potential to generate cells of non-mesenchymal lineages, such as differentiation into neurons or even hepatocytes, the main cells of the liver.
Wow.
Their ability to be derived from various autologous tissues and their excellent clinical safety profile have generated immense clinical interest, particularly in reconstructive surgery.
Okay, so we have the ingredients. We know the power and limitations of somatic, embryonic, induced, pluripotent, and mesenchymal cells. The challenge now shifts completely, right? From finding the cell to actually assembling a functional tissue. Let's talk about tissue engineering.
This is the current frontier. Providing the basic cellular materials is phase 1. The challenge now is phase 2. How do we take these isolated, amplified, and differentiated cells and assemble them into functional, complex tissues or organs that can survive, integrate, and actually work within the human body?
What is the current extent of clinical success? What are we good at building right now?
Clinical use has largely been restricted to cell types that can either self-assemble into a relatively simple structure in vitro or, once delivered, can organize themselves after implantation in vivo.
So simple sheets of tissue.
Think simple sheets like epithelial grafts for burns or relatively simple structural masses like small chondrocytic grafts for cartilage repair. Building a full multi-layered organ requires much more sophisticated tools.
So advanced tissue engineering needs to get beyond simple self-assembly. What are the key tools being exploited to facilitate these more complex structures?
We rely on two primary mechanisms. First, the controlled use of bioactive substances. These are molecules like specific growth factors or signaling pathway inhibitors used to precisely control cell behavior, guiding their shape, proliferation rate, and final differentiation state.
And the second mechanism is providing the physical architecture, the frame.
That's the use of scaffolds and matrices. These can be synthetic polymers or natural biological materials. These engineered scaffolds are absolutely critical. They provide physical support, assist the structural growth of the tissue, and help deliver the cellular constructs in the correct three-dimensional orientation into the body.
So they act as a temporary home until the new tissue structure is stable.
Exactly.
This sounds like complex biological construction. If the ultimate goal is generating a large, complex organ, say a whole kidney or a functional segment of lung, what are the major technical hurdles that still prevent us from achieving this?
There are two showstoppers. The first is simply architectural complexity constructing tissues formed of multiple cell types. Nerves, blood vessels, structural cells, functional cells, all arranged in the correct functional geometry.
Hmm.
But the single biggest hurdle, the one that kills most large constructs, is the vascularization problem, which is tied directly to the size limitation.
Okay, explain the vascularization problem in detail for the listener. Why is it so critical?
If a construct is too large, and that can be just a few hundred microns thick, the cells in the center cannot survive. They can't rely on simple diffusion of oxygen, carbon dioxide, and nutrients from the edge of the tissue.
They starve.
They starve and suffocate. For a large engineered organ to survive and function, it requires a fully integrated functioning blood supply, a vasculature established very quickly. Without this plumbing system, the core of the engineered construct dies.
So how are scientists attempting to solve this critical biological plumbing issue?
Researchers are pursuing some incredibly ingenious biological and mechanical solutions. The biological approaches include supporting the growth of cellularized scaffolds by transplanting them to surrogate hosts.
Surrogate host.
Using another area of the body, often an accessible, highly vascularized site like a muscle pocket, as a temporary incubator to establish blood supply before moving the tissue to its final destination.
And the most complex approach sounds almost like science fiction.
That involves pushing the boundaries of what we call xenogenesis, using highly complex genetic manipulations that allow implanted human stem cells to form organs during the embryonic development of other species.
You're growing a human organ inside another animal.
In essence, yes. This allows the human cells to tap into the perfect developing vascular system of the host embryo. Of course, these approaches are strictly experimental and require intense ethical consideration.
Are there mechanical in vitro advances helping us pattern and support these large structures in the lab?
Yes. This is where advanced manufacturing meets biology. We're seeing rapid advances in three-dimensional cell culture techniques. One key technology is bioprinting.
Like a 3D printer for living cells.
It's exactly that. It uses specialized equipment to precisely deposit cells and matrix materials in a layered, patterned fashion, essentially printing a three-dimensional tissue structure with controlled architecture. We're also seeing the development of novel perfusion bioreactors. These are advanced culture systems that provide better, continuous support for cell growth. They function by constantly circulating nutrient-rich media and removing metabolic waste products, effectively mimicking the function of an early blood supply in vitro.
This all sounds like we are minutes away from having a catalog of replacement organs. What is the crucial reality check we need to maintain here?
The reality check is twofold, addressing viability and cost. First, the application of these highly advanced procedures to the regeneration of large, complex organs remains, today, essentially experimental. We've mastered simple sheets and tubes, but building a fully functioning, durable, multi-component organ is still aspirational.
And the second part.
Second, the cost. Even the successful small-scale procedures require highly specialized facilities and labor. The cost of such organ production is a hugely significant factor that must be drastically reduced before widespread clinical viability is achieved.
Let's pivot now to where this science is already being applied and tested, in the clinical problems of otolaryngology. As you pointed out, the head and neck is a high-stakes, diverse area, requiring regeneration of so many different tissues.
It's the perfect testbed because of that high diversity and complexity required in a small anatomical space. We can look at structural reconstruction, sensorineural challenges, and functional airway replacement.
Start with structural reconstruction, the external ear and the nose. What is the fundamental clinical need here, and what were the severe limitations of the traditional approach?
The need comes from congenital defects like microtia, trauma, or tissue loss following cancer resection. The existing gold standard procedure often involves the surgeon harvesting costal cartilage, that's rib cartilage, which is then carved into the desired shape.
Which sounds painful.
It causes significant morbidity and pain at the donor site, the chest, and often requires multiple, complex, multi-stage surgeries.
And what does the stem cell alternative offer to address this invasiveness?
It offers a complete shift. The alternative uses autologous stem cells, typically mesenchymal stem cells, sourced from a small local biopsy, say, a small piece of nasal septum cartilage. These cells are then expanded in vitro to produce vast quantities of cartilage after being seeded onto an appropriately shaped bioscaffold.
Are there clinical success stories for this technique already?
Yes, there are exciting, aesthetically successful reports. Engineered cartilage grafts, generated in vitro from nasal septum biopsies, have been used for successful nasal reconstructions.
So what's the promise here?
The promise is consistent structural results, dramatically reduced surgical complexity, fewer stages, and significantly lower postoperative morbidity compared to the highly invasive traditional rib harvest.
If you are constructing a large structural scaffold, like a nose or an ear, it needs a perfect integrated covering. How do you handle the epithelial skin layer?
They leverage a well-established technique. The structural scaffolds can be covered in autologous epithelial sheets derived from epidermal stem cells. You take a small skin biopsy, expand the stem cells in vitro into large sheets, and then apply those sheets to the scaffold.
Ah, the same technique used for burn victims.
Exactly. It has been utilized safely for decades for massive skin replacement in burn patients and translates perfectly to covering these new structural constructs.
What about something as delicate as the tympanic membrane, the eardrum?
Augmented repair of chronic tympanic membrane perforation is a very feasible future area. Current therapeutic work has mainly focused on using growth factors and biomolecules to enhance natural healing. But the regenerative potential is high for developing matrices populated with autologous chondrocytes for structural integrity and epithelial cells, providing a combined cellular and structural approach to repair the eardrum perfectly.
Now let's move to perhaps the most ambitious goal in otolaryngology: treating sensorineural hearing loss resulting from cochlear damage. This type of hearing loss is defined by its irreversibility.
This is a monumental challenge. Hearing loss involves the irreversible loss of highly specialized sensory hair cells, the transducers that convert sound waves into neural signals, and/or the associated spiral ganglion neurons.
So the regenerative aim is not just to patch it up.
No, the aim is to structurally and functionally regenerate those lost cochlear structures.
What are the primary hurdles researchers face when trying to rebuild the inner ear? It's so small, so complex.
The challenges are immense and multi-layered. The primary biological issue is identifying stem cell types appropriate for reliable differentiation into sensory and neural progenitor cells. We need to find the specific biological switch to make those cells.
And the secondary issues are purely logistical, related to the microscopic architecture of the inner ear.
Precisely. The secondary issues are highly complex logistical and surgical challenges. Specifically, how to deliver the differentiated cells to the exact correct location within the delicate, fluid-filled cochlea, or, alternatively, how to guide the differentiation process in situ right there in the living ear without damaging the remaining structures.
So is there any evidence that we can actually create a hair cell from a stem cell?
Yes. Feasibility has been demonstrated. Rodent mesenchymal stem cells and other stem cell types have been shown to differentiate in vitro into cells that exhibit the properties and even the specialized structures of sensory hair cells.
And what is the most advanced technique we have to replicate the complex functional unit of hearing?
The truly advanced work involves using murine embryonic stem cells in a highly orchestrated, stepwise in vitro process. This method meticulously mimics the complex, dynamic cellular interactions that occur during normal embryonic development of the inner ear.
And the result?
The researchers successfully differentiated these ES cells into both functional mechanosensitive hair cells, so cells that can react appropriately to mechanical vibrations, simulating sound, and the associated sensory neurons that interact to form specialized synapses.
So we can create the basic listening unit in a dish, capable of receiving a mechanical signal and transmitting it electrically. That is an absolute measure of success in terms of complexity.
It demonstrates that the biological code exists and can be replicated, which sets the stage for future human trials.
Let's move to another major airway structure, the trachea. Tracheal replacement requires structural strength and a functional lining, making it one of the most complex tubes in the body to replicate.
The requirements are almost conflicting. Large constructs need significant structural strength to withstand collapse from the negative pressure of inspiration, and simultaneously, they need an appropriately differentiated luminal epithelium, the inner lining to clear secretions and debris via cilia, preventing infection and obstruction.
The source material highlights two very successful strategies for replacing the trachea. Let's break down the first one, which uses a biological scaffold.
Okay, strategy one involved using a decellularized donor human trachea as the initial scaffold. This means taking a donor organ and stripping away every single donor cell, leaving behind only the protein matrix, the natural shape and strength,
And empty shell.
an empty biological shell. This matrix was then repopulated or seeded with the patient's own cultured cells. autologous epithelial stem cells for the lining, and mesenchymal stem cell-derived autologous chondrocytes for the structural cartilage.
And this newly populated scaffold was grown in the lab before transplantation.
Yes, it was grown within a sophisticated bioreactor to allow initial integration and growth. And this tissue engineering strategy has yielded remarkable, successful 5-year clinical results after transplantation.
5 years is a huge milestone.
It confirms that decellularization plus autologous cell repopulation is a viable long-term solution for complex airway replacement.
That's amazing. But it still requires a donor organ scaffold? What was the second successful strategy that completely avoids the need for a donor trachea?
Strategy 2 represents a paradigm shift away from donor tissue. It involved the construction of a bio-artificial polymeric scaffold. This was a synthetic material engineered to meticulously model the patient's tracheobronchial size and morphology. This synthetic scaffold was then seeded ex vivo outside the body with autologous bone marrow mononuclear cells.
Why mononuclear cells from the bone marrow? What's special about them?
Mononuclear cells contain a mix of stem cells and progenitor cells. Crucially, the implantation of this scaffold relied on the use of growth factors to induce the mobilization of endogenous stem cells.
So you're calling in the body's own repair crews.
Exactly. After transplantation into the patient's chest as an airway replacement following cancer surgery, the patient's own remaining, local stem cells were signaled to migrate and colonize the synthetics. Regenerative medicine here aims to achieve recovery beyond simple scarring. The complexity is huge because the vocal folds operate under intense biomechanical stress. Current research is focusing heavily on assessing various types of mesenchymal stem cells in animal models of vocal cord injury.
How exactly are MSCs believed to enhance vocal cord healing and reduce scarring?
They appear to have a dual mechanism related to structure and signaling. MSCs support vocal cord remodeling and actively reduce scarring when they are directly injected, either alone or within a composite scaffold.
Right.
When injected using hyaluronic acid hydrogels, the gel provides immediate volume of filler, while the MSCs provide the biological activity, leading to accelerated and less scar healing in small animal models.
That immediate volume and the biological kick sounds promising. But given the high-stress environment, the constant vibration of speech, does the clinical uncertainty about persistence hold up in human applications?
It remains the critical clinical uncertainty. Vocal fold remodeling and maturation can take up to 6 months in humans. So researchers are actively working to determine whether the injected MSCs or their derived factors persist long enough in that high-stress dynamic environment to provide a sustained long-term benefit.
And how about replacing the specialized covering of the vocal cords?
The vocal cords are covered by a delicate, non-keratinized epithelium, a mucosal lining that must remain pliable and moist. This can potentially be replaced using 2 methods. First, by the in vitro expansion of stem cells derived from small biopsies of adjacent healthy mucosae.
And the second?
The second is using that advanced iPSC technology we talked about. Scientists can differentiate human-induced pluripotent stem cells in the presence of specific matrix molecules and growth factors to generate that precisely specialized non-keratinizing epithelial lining.
That opens up a huge avenue of hope for people who rely on their voice professionally or simply want to restore normal communication. Before we transition to malignancy, let's quickly cover peripheral nerve and large bone regeneration in the head and neck.
Peripheral nerve damage is a common complication following head and neck cancer surgery, often affecting the facial nerve or the recurrent laryngeal nerves, the RLNs, which control the vocal cords. Research shows promising enhancement techniques. For RLN injuries, muscle-derived mesenchymal stem cells have been shown to improve functional outcomes.
And for those devastating facial nerve injuries.
Neural regeneration following facial nerve injuries is enhanced by a powerful combination of 2 elements. Neural-induced mesenchymal stem cells, so MSCs, that have been pre-treated to lean toward a neural lineage, and platelet-rich plasma, or PRP.
Which is a concentrated source of the patient's own growth factors.
Exactly. This combination appears synergistic in promoting faster and more complete functional recovery.
You also noted that the head and neck region itself is a rich source of specialized stem cells.
Yes. For example, certain regions of the nose contain olfactory and sheathing cells. These cells naturally guide nerve growth in the olfactory system. They act as a source of autologous stem cells that researchers are now using to assist neural and other regenerative procedures, essentially leveraging the specialized biology of the region.
Finally, large deficiencies in the maxillofacial skeleton, the bone structure of the face and jaw from trauma or cancer resection.
This is a prime application for MSCs. Regeneration of these large bony deficiencies is effectively assisted by the direct injection of MSCs combined with appropriate scaffolds, such as ceramic or polymer materials, which guide the MSCs to differentiate into bone cells.
On scars.
Furthermore, stem cells, particularly MSCs, are being extensively investigated for improving the quality and appearance of scars following surgery or trauma by reducing fibrosis and promoting organized tissue remodeling.
We've established the incredible potential of stem cells for healing and regeneration. Now we have to turn to the other side of the coin, the dark side of stemness. The critical role of these cells in tumor growth, invasion, and metastasis, otherwise known as cancer stem cells or CSCs.
This transition is vital because the same biological controls we seek to master for regeneration are the controls that go rogue in cancer. This is a clinically critical area. If we look at head and neck squamous cell carcinoma, or HNSCC, which accounts for the vast majority of head and neck malignancies, the statistics are sobering. Only about 50% of patients survive longer than 5 years. Standard therapies often fail.
And what does research confirm is the driving force behind this persistent treatment-resistant growth?
Recent extensive work confirms that tumors contain specific subpopulations of altered stem cells, which we term cancer stem cells. Crucially, tumor growth, recurrence, and resistance are ultimately dependent on these cells. This pattern has been identified universally, both in squamous cell carcinomas like HNSCC and in glandular tumors such as salivary gland mucoepidermoid carcinomas.
These cells have a normal origin, but they've been hijacked. What are the unique properties of these cancer stem cells that make them such a clinical nightmare and distinguish them from the bulk of the tumor?
They possess 3 unique properties that directly undermine standard cancer therapies. First, extreme resistance to therapy, both chemotherapy and radiation. Second, an enhanced ability to invade locally into surrounding healthy tissue. And third, they possess the innate ability to metastasize.
Spreading to distant sites.
Exactly, spreading to distant sites like the lungs or liver, which is the event responsible for the vast majority of cancer patient deaths.
Let's delve into the process of alteration and dysregulation. How does a normal, well-behaved stem cell become a malignancy-driving CSC?
The process is complex, driven by both internal and external insults. We know mutational effects are key, often driven by exposures like tobacco and alcohol. Crucially, we are seeing a massive increase in oropharyngeal tumors associated with human papillomaviruses, or HPVs.
And what do the viruses do?
The viruses don't just cause a lump, they actively increase the number of normal stem cells and severely alter their behavior, pushing them toward malignancy.
So it's a failure of the body's normal control system, a loss of homeostasis. How does that look at the signaling level?
Normally, stem cell divisions are perfectly balanced. Some divisions renew the stem cell population, and others produce differentiating cells that mature into functional tissue. In malignancy, key signaling pathways like Notch, Wnt, and sonic hedgehog become dysregulated.
There's a glitch in the system.
A major glitch. This biological glitch shifts the division pattern of the cancer stem cells almost entirely toward greater self-renewal.
Meaning they stop differentiating into helpful mature cells and instead just churn out more malignant stem cells, driving the tumor's growth and complexity.
Exactly. This leads to an exponential increase in the number of CSCs and the complete loss of control over tumor proliferation.
Beyond just replicating, CSCs demonstrate superior defense mechanisms. They survive treatments that kill 99% of the surrounding tumor mass. How do they achieve this enhanced survival and resistance?
Normal stem cells are intrinsically robust. They have enhanced mechanisms to maintain their cellular integrity, which is necessary for their long-term function. In cancer stem cells, this mechanism is pathologically enhanced, resulting in significantly greater resistance when they are challenged by cytotoxic agents or radiation.
We need to understand the radioresistance mechanism specifically. The source material notes it is associated with altered levels of reactive oxygen species.
Right. Radiation therapy works by flooding the cells with DNA-damaging molecules, specifically reactive oxygen species, or ROS. ROS are highly reactive molecules that cause irreparable damage to the cellular machinery. The association suggests that CSCs have developed enhanced systems, far superior to normal tumor cells, to effectively manage, neutralize, or detoxify the ROS. By efficiently detoxifying the radiation damage, they protect their DNA and cellular machinery, securing a massive survival advantage.
That explains their resilience, but not their ability to spread. Metastasis is the primary cause of death. How do these normally tightly packed epithelial cells manage to physically break away and move through the body?
This is where a process called epithelial mesenchymal transition, or EMT, is implicated as the crucial mechanism of escape. In this process, cells that are normally tightly cohesive epithelial cells stuck together acquire specialized mesenchymal characteristics.
What are those key mesenchymal traits?
They undergo fundamental changes. They lose their mutual attachment to neighboring cells, they change their cytoskeleton and shape, and they become motile. They stop acting like building blocks and start acting like mobile invaders.
And they use this to escape.
The CSCs utilize EMT to escape the primary tumor, migrate through the bloodstream and tissues to distant sites, and then, and this is the kicker, they undergo the reverse process, mesenchymal epithelial transition, MET, where they revert back to the proliferative epithelial phenotype to produce a secondary metastatic tumor.
So they switch gears for the journey, and then switch back to a growth phase once they arrive at the new colony site. It's an incredible level of functional plasticity.
It is, and the extent of this EMT-MET switch is critical. It depends partly on factors intrinsic to the tumor, the genetic makeup, but also critically on extrinsic stromal factors.
Signals from the surrounding tissue environment.
Exactly. Signals like specific cytokines and local conditions like hypoxia or low oxygen levels. These external factors can trigger or suppress the metastatic switch.
Given that eliminating cancer stem cells is absolutely necessary for curing solid tumors, what are the primary therapeutic strategies being explored right now to target them specifically?
Researchers are developing a kind of three-pronged approach aimed at their unique vulnerabilities.
Let's start with how to find them among the mass of non-stem cells.
That's the first approach. Targeted delivery. This relies on the fact that CSCs express unique cell surface markers. For example, using specialized antibodies against specific markers like anti-CD44V6 bevacizumab Mertensine researchers can use the marker as a homing beacon to deliver highly potent cytotoxic agents directly and selectively to the cancer stem cells, sparing the rest of the body.
The second strategy attacks their source of immortality, their enhanced self-renewal.
Correct. The second strategy is inactivating self-renewal. This involves developing small molecule drugs designed to specifically shut down those dysregulated signaling pathways, Notch, Wnt, etc., that promote infinite replication. A specific example is a drug targeting the Notch signaling pathway, a Notch2, Notch3 antagonist called tirextumab. By blocking this, the tumor loses its engine of self-renewal.
And the third strategy addresses their exceptional resistance to our current best treatments.
The third is reducing resistance. This involves identifying and inhibiting the factors that protect the CSCs from radiation or chemotherapy. One promising target is inhibiting factors like connective tissue growth factor, which has been shown to activate pluripotency genes in head and neck cancer cells. By inhibiting this factor, we reduce the CSC's ability to maintain that robust, resistant phenotype, making them more vulnerable to standard therapies.
All of this sounds highly promising. But you mentioned a critical final biological challenge regarding CSCs and chemotherapeutic drugs, their plasticity.
The plasticity is what makes cancer stem cells the ultimate challenge in oncology. They are not one static population, they are fluid. They generate cells that differ vastly in their drug or radiation responses, and they're able to switch back and forth between functionally distinct phenotypes.
What are those different functional identities?
Research shows they can switch between a preferentially migratory phenotype ready to undergo EMT and spread, and a proliferative phenotype ready to aggressively grow the main tumor. They essentially have a traveling identity and a growing identity.
If they can switch jobs, how can we possibly eliminate the entire population? If we target the proliferative cells, the migratory cells might survive and spread.
This functional flexibility means that successful, comprehensive elimination of the entire CSC population may absolutely require drug combinations. We need combinations that simultaneously target both the migratory forms and the proliferative forms. Furthermore, this necessitates the urgent development of new assays, new tests that can assess the drug effects on each of these phenotypic manifestations simultaneously in the lab, so we know exactly what combination to use to hit them in all their identities.
This has been a fascinating and incredibly detailed deep dive, covering both the regenerative potential and the malignant threat posed by the same core cell type. Let's synthesize the key takeaways.
At the highest level, the knowledge we've gained confirms a fundamental biological duality. Stem cells are the ultimate origin of all subsequent tissue growth, maintenance, and complex regeneration, providing the foundation for our most ambitious regenerative procedures.
But on the other side,
Conversely, These same cells, when altered, are responsible for the accelerated growth and devastating treatment resistance of malignant tissues.
The good news is that recent research has clearly illuminated many aspects of the control mechanisms underlying stem cell behavior, whether normal or malignant. We have begun to master the biological switches.
This knowledge is being translated into profound clinical action. It's being used to develop revolutionary clinical tissue regeneration methods, like building a new trachea or growing functional, sound-sensitive hair cells, while simultaneously driving next-generation cancer therapies aimed squarely at eliminating the incredibly resilient cancer stem cells.
The mastery of stem cell biology offers a fundamental shift in medicine, moving beyond simple repair to the true regeneration of lost complexity, from the delicate architecture of the cochlea to the precisely biomechanical vocal cord.
Yet the simultaneous evolution and plasticity of cancer stem cells means that our understanding of these fundamental biological controls is essential for both regeneration and the long-term survival of the patient. The battle for the body is fought at the level of the stem cell.
That leaves us with a provocative thought to close on. We have learned how to use scaffolds, potent growth factors, and genetic prompts to guide stem cells. We've also learned that external signals, the extrinsic stromal factors, the mechanical forces, the low oxygen conditions, can profoundly influence a stem cell's fate. What unexplored aspects of the tissue environment might we manipulate next, not just with drugs, but with environmental cues, to tip the balance decisively away from cancer progression and completely toward perfect functional tissue regeneration? That ultimate biological switch remains the key to mastering both life and death.
A profound and ongoing question that drives thousands of researchers globally every single day.
Thank you for joining us for this deep dive into the complex world of stem cells and regeneration and malignancy. We hope you feel thoroughly informed and ready to follow this astonishing field of research. We'll see you next time.